Okay, hello everyone. In case you could not come to see my poster, and I will record this video for you. And hello everyone, and I'm Huan-Chuan, and this is submission number 341. I think the category is W1. Display information is 24/7 and 12. Okay, so title is developing an adjustable workflow for machine learning supporting glycan annotation on LCMS MS data. So let me have a brief introduction about glycosylation. It's a kind of post-translational modification, just similar to like phosphorylation, ubiquitination, methylation, etc. So you can consider it as a modification acting mainly as a signaling molecule. And this kind of PTM, post-translational modification, is well regulated and for like protein activity or even some biological function as well. So here, glycosylation especially means those carbohydrates, uh, molecules modifying mainly on proteins. So maybe they could somehow it could be as a free form or uh, linked to lipid. That's possible. And about the roles of glycosylation in our body, I could say that mostly we learned that glycosylation on proteins could be recognized as a signal for many physiological function. I think, yeah, those like in development and also for immune system and even like your body interacted to other microorganisms like bacteria or even virus. And there are some types of glycan that maybe not carrying on cell surface, but they also have some important roles in our body. And let me explain that why we are going to this glycan analysis and why it could be so difficult. First of all, glycogen has varieties in evolution. That means some genes are shared to make same or similar glycan motifs, while some species family they developed their own specific motifs. So some glycan motifs could be general, universal, or some could be species or even families specific. And also, there are another issues that is glycophone varieties. That means each site carrying glycans are not fixed. It could be dynamic and based on the conditions and the gene expressions that will modify the dynamically change the glycosylation on certain protein glycan carrying site of a protein. So here's an example that is SARS-CoV-2 spike protein, and I think you can see that there are many glycan carrying site that has multiple possibilities of glycans carrying on it. So that means if you get like 100 of the protein, glycoprotein fragmentations ions, you may get like half or like 70%. I don't know. Like you can see some types of glycosylation on certain sites. So we could not definitely tell you that 100% that one site has this kind of glycans, but we can tell you that if it is possible to find this glycan on certain sites. And another more likely technical issue is that we usually only get limited amounts of glycans in real samples, leading to insufficient numbers of annotated spectra. That's because like we after we purify the glycans from glycoprotein sample, we still have lots of different types of glycans, and those more abundant glycans could be in a range that is detectable by the instruments, while those minor ones could be in a very trace amount, like maybe less than a nanogram. So divided by the molecular mass, you have less than picomole or even femtomole. So that's very difficult for current mass spec instruments. Maybe it could be solved in near future. Yeah, and then after we could obtain those high quality glycan containing spectra. We still need an expert to annotate and solve the spectrum itself. So this really takes time, and not everyone could easily do this job. Yeah. So the workflow to here we acquire and validate data in glycomic analysis, and could be like this figure. So we have certain context, topic, something you want to investigate. You could find information from papers first, publications, and if there are some gene or protein expression data online or by experiments you have done in your lab, you can construct that predicted glycan list that based on the evidence, informations, and at this moment, you don't know that if the glycans really present. So you need to do the experiment to analyze the sample following your your condition. And after you getting those spectra from mass spec, you need to view the spectrum and search if those glycans are presented in your spectra file. And then if you find it, you can list those glycans, and if no, you can exclude those one, and finally do some statistic like calculate glycan abundance, or if you are certainly you want to compare the terminal epitopes, yeah, you can do some quantitation and see the result. If a result is consistent to your hypothesis that glycan profile changes due to maybe your treatments or something. Yeah, and I think there are three very difficult barriers that is very difficult to solve. That the first one is the format is not compatible. I mean, it's like waters or thermal features, they have different file formats, and when you reviewing the spectra is very time spending, and also maybe you are not in the good condition tomorrow, or maybe you could do the annotation better than your annotation yesterday. So the quality of your annotated data varies by your condition. I mean, sometimes you make mistakes, and maybe you are not in a good mood, or you got you caught the code, so or you are missing some information and you could not annotate data very well. So following these issues, I think is very we have the need to develop something that could help the annotation process more stable, and even more time saving in a time saving manner. So here's our purpose of our the software. We call it Geico mass-based parser, and the workflow here. Yeah, you may have seen it in other conference last year, but yeah, we have changed a lot since that. So first, we still have spectral data you get, and you still need to have some uh, knowledge, informations of your sample before you do this. But we can automate it using this workflow. So let's see. So after you get your spectral file, you still have to do I think for one sample it could be once or twice doing manual annotation, and also we extract your spectral data. So you will have one annotation sheet with your manually reviewed results here. And also there are one extracted MS2 spectra that only contains spectral information here, not specific to glycans or something. And following that, we have some criteria using those so-called feature selection here. And I could say that we base on characteristic fragmentation ions here, and these features could be changed based on different experimental method in your lab. So generally, it's what those ions you often observe in your experiment. And using this, we can first quickly, robustly remove some non-glycan spectra. I could not say it's 100%, but it's like highly possible that it's not a glycan because they don't have those fragments, So we filtered, we list all those non-glycan spectra, and we can have a less file size of probably glycan spectra file. And this spectral file will be combined to annotation sheet following the glycans you found in here by yourself, and we will combine the MS2 information to the specific scan number of glycans you found here. So now you have not only annotations, you also have entire pick list for that icon. And also we will help you filling some, if there is some missing values that's not very critical for annotating. And also we will do some quality control that if you have like less than 5 or 3 counts of certain glycans, this type of glycans will not be able to enter the training model process. And also we have some normalization of your pick list. So after that, you will get one preprocessing dataset. And this process could be automated once you have your feature defined it. So like you have one sample with 10 treatments, and you already decide what kind of glycans you are going to see. So you can decide the features. You can do the this kind of process like 10 times, and you will have 10 different but similar data in one set. We can combine it, so you will get like a database like project management method here. And after that, we could start our training process. So first we use Weka to explore, to mine if there's any possible machine learning models here, and we test as small as possible for our data formats. And also we have some evaluation methods such as clustering to see if the result is correct, or visualization, and also F1 score, much important. Yeah, and we also add some tapes here to get our performance better. Yeah, if you are interested, you can come to contact me or ask me at a venue because I think those details are we will keep those details confidential until the manuscript is accepted. Yeah, but what we could say here is we also have some like stratified cross validation and some optimization to let our data more easily to be to become a training data of a machine learning model here, that makes enable everyone to train your own data sets on your personal computer. So you don't need you don't need a supercomputer or server to run this. So here's our goals. We would like to provide an adjustable solution for of glycomic analysis here with a machine learning pipeline to each lab. So certainly we want everyone could have their own model trained for their lab, and this model performs well. Maybe only on your lab's data, but anyways, it helps you to just like you have another assistant in your lab helping you to do these tasks. And also we want to make files for this kind of analysis more compatible even to multiple devices. Like you can convert file in your lab PC, and you can do the machine learning test at home, or maybe you are on a conference and you have your laptop there. Yeah, and you want to show some results quickly, very quickly. And also we want those files to be compatible in across different platforms such as Windows and Mac OS, and even maybe Linux if if we can, if you can. And also we want to make files more possible in a reasonable file size, so you can share and you can easily reproduce the results from other groups. So like if I say that, yeah, I have glycan, this kind of glycans in my dataset, you can just check check the file and run the models on your PC and check and reproduce the results and see that if I am lying. Yeah. And also here we would like to provide we want to provide criteria of people that when people are creating glycans annotations sheet, we want them to follow certain criteria, so you can make those files how to say that, it's more universal. And I mean, if the data format, if a annotation sheet is following certain rules, and we can use some programs, use some codes to extract the information. So that's the key to automate the analysis workflow, I believe. Yeah, and the ultimate goal of this project is we want to increase number of high quality annotated glycan spectra. So following this, I think you can have better performance in those popular large scale machine learning model or even deep learning model, and also self-supervised learning models if you want. Yeah, but first of all, I think we don't have such a high quality annotated data sets here at this moment. So I think our tools could be the help to increase those high quality annotated spectra. Yeah, and we also have wikis and documentation. So because I believe that, like me, I'm not an expert. I'm not expert. I'm just a beginner to programming. So I think it's very important to have documentations that telling people how to use our tools, and also if there's anything you can adjust, or if you are using correct functions here, if you are using our tools correctly, properly. So yeah, that's really important, and we will make it public, maybe on Notion itself or on GitHub. Yeah, we will decide it when we are about to finish it. Okay, so there's some more detailed parts about how we build. How is this workflow working like? So first one is data exploration. And as I said, you still have to view the spectra and annotating it. Here we said it could be in Excel, but I think anything that acting like this, like if you are using Mac OS, you can use numbers, I guess. Anyway, if you can build a CSV or Excel file, it could be fine. So here we want to ask people, researchers follow certain criteria to build the annotation sheet like this. And actually, I think the key here is that we will ask you structures, and yeah, you can also have a more detailed annotation, or even very precisely describe these glycans here. Yeah, but we leave it blank here because we are still testing. Yeah, so you have actually this annotation and this composition. Yeah, and you have a MS2 scan, and this kind of information is additional data. So yeah, if you have this kind of data, I think it's better for further clustering task, but it's fine if you don't have that. So generally, you will have a MS2 scan, and you will have a data link for it, and some nodes if you want about this spectrum. So if you know the MS2 scan number, we can extract the whole spectrum info from your file and combine the peak list and the intensity into this entry. Okay, so the second one is feature selection and data preprocessing. So here we want you to select proper features, and I could just say we selected some characteristic fragmentation ions, and actually this part can be customized. So I think finally we will have you your own lab need to build one optimize feature sheets for your lab members, but I think is I mean it's not limited to you, but all of other lab members using same protocols to get guidance. And also, like if you are analyzing permanently tainted guidance or even native guidance, yeah, you can edit the fragment ions features sheet because the fragment patterns are different. Yeah, and following this, you need to do data preprocessing, and here we have some default parameters for you, but you can customize it in your need. If you have enough data amounts, you can set like higher lower limits counts for each class of guidance before training. And also here it's okay that if the annotated spectrum don't have the values of certain fragmentation ion signal, yeah, we will fill it with 0, so don't worry. Yeah, and after this we will do a normalization here, and if you are going to train your data in certain models, maybe you may need to change your data type. Don't worry, we will help you to configure this part. And here we want to say that to overcome the issues of insufficient count of minor glycans, we have strict rules to avoid any artificial effects here. So first, we don't have data augmentation. That means because in some machine learning field, like if you're doing an imaging training, you can just do some tricks, maybe rescale it or cut it or like rotate it, and you still have a same annotation of that data. But for our mass spec spectrum file, I think it is not practicable. So we don't have this data augmentation, and also we won't include any in silico data. So yeah, in your own lab, you only include the data that you have seen it. Yeah. And also we allow people to combine datasets from different batch or even specimen, but only if you are doing on the identical experimental protocols. That means you couldn't combine and positive mode or and one negative mode datasets together because they don't have same fragmentation patterns. So you have to train them uh, uh, individually. Okay. So here's the next step is model constructions, comparison, and evaluation methods. So yeah, in beginning we use Weka for data and model mining. And I think one very important point is that our preprocessed file is able to subject to sending to Weka because in Weka I think I don't know if we can really solve these issues, but yeah, if your file is too big, like 2 GB, is impossible to mine the data here. And our file is like for each experiment, one spectral data set is like 100 to 200 megabytes, so it's still possible for data mining and model training here. And we have some model comparison, and we saw that some models performs better, and here we could say that random forest seems to do better than other methods. And yeah, you can discuss with me if you are interested why this method does better. Yeah, and also we have some evaluation method like F1 score, because we have a annotation sheet, so we know the at least is ground truth for the one who annotates who annotated the data. Yeah, so if you are doing this wrong, the result will be wrong, just like you. So it's very important if you like PI professor helping you to make sure you are annotating right. Yeah, and here we can also compare that glycan classes founded. Like if you have 30 glycans, 30 type 30 kinds of glycans, and they have 25 different glycans that over, like they have all more than 5 spectra. And you can see that if all of these spectra glycans over 5 spectra you annotated does do appear in the results. And also I think based on this method we have the ability to figure out if you missed out some spectra. It should be certain type of glycans, but you didn't notice that, or even some glycans that maybe you are too tired, you mislabeled it, or you kid when you entering the value, you mess it up. Yeah. So I think in following our protocol, you could figure out that those missing parts in your data. So what's the feature and what we want to achieve in our pipeline, our workflow here? Yeah, we try to we are trying to achieve nearly perfect prediction results on small scale, well labeled datasets. I mean, it only works for you, and maybe it's performing very bad in other contexts, like in another labs doing analysis in a different way. They have different methods, so maybe your model isn't applicable to them. But for you if you are doing using the same protocols to get your data, I think the model will do well uh, on your data sets. So here, if we can achieve like this kind of perfect, nearly perfect prediction, and in seconds or even minutes, yeah, it can reduce the time you need to search and validate those non-glycan spectra. And also, yeah, you can get those non-glycan quickly using, if you have already trained a model, you can have even those special file that without annotation, and you just send it to the model, and it will tell you that, yeah, those glycans you have seen also present in this data. So you can save time, and you can get more time to validate the results, or even there are some new glycans that you haven't registered to your own model. So yeah, you can focus on those more important and those more minor glycans, I think. Yeah, and the feature, I think a very powerful point is that using these kind of small scale models enable researchers to build models rapidly. So you have some data sets, and you don't need to train models in a supercomputer for days, but you only need to process this data well, annotating them well, and doing this many just time of brewing a cup of coffee. Yeah, and also I think our workflow has many checkpoints for you to review, and because the checkpoint sets are showing you the data that you can uh, confirm it, validate it through your eyes, so you can easily figure out if your settings, if your configurations are wrong, or if you want to change something during the whole in the whole analysis workflow. And you can also share this kind of like intermediate information, or even share your settings to other groups, telling them that yeah, this settings could work. So here's the some preliminary results comparing with other similar solutions. So firstly, we have analysis time spent using a quite old desktop PC. And here, the red highlighted one is our tools, and the blue ones are others different softwares I could get here and could run it on my PC. So you can see that the first step of conversion took not that long, and but the annotation and the analyze time is quite short. Yeah, this less than 10 seconds includes a small scale model training process, and even until you get the results here. So that means if you already have a trained data set, have a trained model, yeah, uh, adding data sets into your model and check the results would be maybe less than 1 second, based on the amount of the guidance had been adding to your model. Yeah, but it's very fast comparing against other methods. So the total time spent here is like nearly 5 minutes, and you can get all of your results here. While other solutions apparently take more time here, if you mind. And also here, I know there is a model that published this year in July, and I wanted to do like comparing a F1 score, but they don't have such a result output for me to compare. So here I only show what I can do here is comparing types of glycans we found here, and we can see that their methods Candy Crunch that they've captured mostly truncated hybrid biantennary, triantennary, tetraantennary, and non-ciliated here N-glycans, and like they couldn't figure out more than 2 cilic acid, while our methods, because we have like this kind of glycans, if we have more than 5 spectra available, yeah, it could find it out from your data set here. So the glycan types that you can see that can decode from 41 different glycans, and here our method found 42, and there are just only 16 glycans overlapping here. So I will say that yes, we can have more glycan overlapping here if you also manually speculate. If you annotated those kind of glycans here, yeah, I think our methods would help you to to find it out. Yeah, because I didn't annotate this kind of truncated or like not that difficult not that more complicated structures. Yeah, I would like to check more complicated glycans that has more glycans that have a more complicated terminal structures here. So yeah, we're dedicated to this kind of glycans, but this kind of more not complicated glycans here. So theoretically, I think our method could fully cover this model if you really working hard on annotating all of the glycans you have. You could see here. Okay. So I think that's all, and here's some future work. We want to support more raw spectral data formats. So yeah, we still currently only support thermal features raw data. And we want to add more models and tutorials on our site, and also make some GUI graphic user interface, so you can just by clicking, dragging, hitting those files, and everything could be there. And also we would like to combine with our tools with other solutions to make a better effect here. Yeah, that's all. Thank you for listening.